Okay, welcome to the first lecture on sequence alignment. This video will be about pairwise sequence alignment, and you'll want to download the PowerPoint from the alignment module on Canvas. Okay, so if we were going to continue on with algorithms, the next three things that we would go over are the sort of algorithms that underlie pairwise sequence alignment. Up to this point, what we've done and where we finished with the Rosalind problems was finding short strings, or what we call patterns or motifs, k-mers, short sections of the sequence, and then we could search for how many times those things occurred in some larger text based on a certain number of mismatches that we wanted to allow. So in some sense, that is doing sequence alignment, but as you'll see in this lecture, the problem becomes substantially more complicated when you want to align two full sequences and you want to make sure that you're getting the best alignment across the whole sequence, because that involves not just matching the sort of pattern, right? You could, at least in principle, think of just take one sequence and use it as the pattern and then slide it along and see if there's a match in the, or get the Hamming distance, for example, compared to the other sequence. Well, that doesn't always give you the best alignment, and especially if you want to allow for the fact that there might be insertions and deletions in that sequence. So that's what we're going to get at today. Even though I haven't assigned Rosalind problems for these algorithms, they are available for you to answer. You can just look up the textbook track in the Rosalind database and find the problems there that sort of go along with algorithms. And if you have trouble finding that, feel free to reach out and I can point you in the right direction on those. Okay, so what is the alignment game? What makes doing alignment so hard or so different than what we've done up to this point? Well, if you think about the two, let's see, I just want to have a better pointer here, right? If you think about these two things as our two sequences that we want to align, the objective is going to be to come up with some kind of metric that guarantees that we're getting the best alignment of these two things. So a very simple scoring system would be to give it a score of 1 if there's a match and a 0 if they don't match. And so if they match, we'll just take off that first letter in the sequence and move along. Otherwise, if there's not a match, we can consider whether we ought to take a symbol off of just one of the sequences. So what does that look like? So first position scores a 1, it's a match. Second position scores a 1. Third position, it was a mismatch. So let's consider whether we can move it along. Now, obviously I know the solution here, so I'm not going to go through all the potential things you might do to maximize the score. But let's say we add a gap here so that then we score a match there and there. Now we have another mismatch. So we slide it along, don't get any better really. Now we're not going to get any better, so we might as well score these as mismatches. And in this scoring scheme, mismatches don't really count any different than a sort of non-alignment where we inserted a space. So these dashes are like insertions, and we'll call them indels. I'll talk more about that along the way, but you can't tell, for example, that whether this T is an insertion or there's actually a deletion in this sequence. There's no directionality in terms of gain or loss of that base pair. When you only have two sequences, you just can't tell. Okay, and then we might do that. Okay, and in the end, there are only 4 matches with that sequence. And because, you know, this is my example and I'm kind of cheating, I know that's the best solution given these 2 sequences. Okay. So this is just what I was saying about the polarization of knowing insertions and deletions. I've color coded them, but that it's a little arbitrary which string you're calling insertion and which one you're doing deletion when you only have 2. So one way to think about this, and a lot of this is going along with, you know, trying to help you think about if you are going to go ahead and work these out, do the code to write these algorithms. What you're doing is treating these like a 2-row matrix and then comparing in the matrix where do you get pluses and minuses and where do you have to put in the dashes. Right, so you've got matches, insertions, deletions, and mismatches that all need to be accounted for. So one of the considerations when you're doing an alignment is what is the longest common subsequence, right? Because we might want to say that we really don't know what's the best alignment out here in those last 4 characters, because no matter how you slide these around under this current scoring scheme, you're going to get the same score. But over here, the best alignment of this half is 4 if we just put an insertion at the C. Okay. Now, obviously, again, I cheated with this example, kind of knowing the best alignment to begin with. But the reason that's tricky is that if you were coding this, the computer doesn't know what the best alignment is. And so really to get the best alignment, it would have to compare all possible alignments, which means adding dashes to all possible positions. And so that gets to be a really big number. So when you're just, again, we're just doing pairwise alignments, 2 sequences. 2n, choose n. That's, you know, if your sequence, right, if n is the length of the sequence here, that gets really big really fast, right? 2n factorial over n factorial squared. There are a lot of possible combinations when you start to get to anything like a gene or let alone a genome sequence. It pretty quickly becomes computationally not doable to sort of brute force, which is the way I showed this, the best possible alignment, because you just can't do all the combinations. So the Roslin problem that goes along with this would be to find the longest common subsequence of 2 strings. So you're basically looking for a subsequence of that gives you the highest score. But we're not going to do the programming for these. So one of the sort of intermediate steps that I want to talk about here is and it's sort of interesting in a historical aspect that the alignment game, you gain a lot of sequence from mapping problems. And so the Manhattan Tourist problem is kind of a famous math problem where the objective is to go from some point in New York City to some other point in New York City and see the most attractions. But you can only turn south or east. Okay, so you can't backtrack. You just want to take the route through Manhattan that lets you see the most attractions. Okay, And you'll see in a minute why this is related to the alignment problem, because really what, just to sort of let you know where we're headed, you can think of this grid as our two sequences, right? The sequence 1 provides the columns and sequence 2 provides the rows, and we want to find the track here that makes the most matches or optimizes the score for the alignment between the rows and the columns. Okay. So if we translate our sort of map of Manhattan here into this grid where we just put the numbers on each road or each block, depending on whether they have an attraction or not, the problem then, because we've abstracted a little bit, maybe becomes a little more obvious. We just want to find a route through here that optimizes the score, and that will give us the route that tells us which route lets us see the most attractions. So here I've expanded the grid, simplified it a little bit. Expanded, I mean made it bigger, but there are actually less nodes in it. And we've added weights to each of the roads to just illustrate, for example, how many attractions are there. These are called the weights of the edges. So each of these arrows is an edge in the graph, and each of the dots is a node. Okay, and so our Manhattan tourist problem is to find the path from the blue circle to the red circle that optimizes the score. Okay. Okay, so that's just the Rosalind statement of this problem. How do we go from the source to the sink in the grid and get the highest score? Okay, and so the first sort of most simple way to think about doing this problem is called a greedy algorithm, right? So we just start at 0, and whichever path gives us the higher score, that's the path we're going to take, right? So if you're standing at the corner of 8th and 59th Street, and if you go east, you're going to see 3 attractions, but if you go south, you're only going to see 1. Well, then, hey, let's go east and we'll see 3 attractions. So then we give that node a value of 3 because that's how many attractions we've seen, and we just ask the same question over and over. From this node, if I go east again, I get 2, but if I go south, I don't see any, so I'll go there. Okay, and then so on, right? Now we've seen 5 attractions. Should we go east or south? Well, we should go east again, and then south, right? So same decision process here. Here was sort of arbitrary. We could have gone that way to get 2 or south to get 2. I arbitrarily chose to go south. 3, we'll get to 19, 1, because we only have the option now to go south. Okay, and so we saw 23 attractions. Now the question is, and for example, there was only one choice here where it was an arbitrary decision. If I had gone east here, I would have to 15, 16, 17, 18, 19, 20. I would have had a lower score. So in some sense, that arbitrary decision actually worked in our favor to see more attractions. But the question sort of is still out there because we took this greedy approach where at every decision point we picked the one that gave us the most. Is 23 attractions the most we could have visited under the given conditions of only going east or south? So is the greedy solution the optimal problem? Well, obviously with all that lead in, you might suspect already that it is not. Okay. One of the more famous illustrations for why the greedy algorithm doesn't work is the change making problem. So the change problem is finding the minimum number of coins needed to make change, right? So intuitively, we do this in our head all the time. And so think about how you do it. Your brain essentially uses the greedy algorithm to solve the change problem. You basically have an amount of change that you need to get. You think, okay, what's the biggest coin that I can choose to sort of fill my need? If I need to give 30 cents in change, right? I'm going to pick a quarter, so give them 25 cents, and then now I have 5 cents left over. The biggest thing that fills that is a nickel, so I'm going to give them a quarter and a nickel. You just used the greedy algorithm to solve that problem. That works okay for the denominations of coins in the United States. That does not work everywhere. Okay, so this is money changing if we were in Tanzania. So these are you've got a dollar coin, a 50-cent piece, a 20-cent piece, 10, 5, and 25 cents. Okay, so in the US, right, if we were doing 25 cents, if we had to do 40 cents, we would do a quarter, a dime, and a nickel. Okay, and that works. That would be the solution. That is the most efficient, that is the fewest coins we could give the person to get 40 cents. But by Tanzania's coinage, we could give them 2 20-cent coins, and that would be better. The greedy algorithm won't work to give you the optimal solution in Tanzania, right? Because you would give them 25, 10, and 5, just like you would in the US using the greedy algorithm. They have a 25-cent coin. So you would give them those 3 coins when there's no sort of way in the greedy algorithm to recognize, oh, hey, actually, I ought to give them just 2 20-cent coins instead. Okay, so because there aren't that many factors to keep in mind when you're making change, this doesn't maybe seem like a super hard problem, but when you extend that to the Manhattan tourist problem, it definitely should cast doubt on whether taking the greedy solution every time is going to give you the most attractions visited. So if we sort of tear that problem down a little bit, one solution, one possible solution is called a recursive algorithm. And this will give us the right solution, even no matter what the denominations are, right? So in this problem for Rosalind, the denominations are arbitrary. The example here, we need to make 9 cents change, and the denominations of coin are 6, 5, and 1. So the recursive algorithm says minimize the number of coins. In this case, we need to make 9 cents change, but you need to compute what's the minimum number of coins if we start with a 6-cent coin. So from 9, if we use first a 6-cent coin, we're going to go down to 3 cents left over. If we use a 5-cent coin from 9, we're going to end up with 4 cents left over. And if we do the 1-cent coin, we'll end up with 8 cents left over. Now, what we can do, remember, we used recursive algorithms already. We were introduced to those in the pattern to number and number to pattern in how we sort of generate the index values for a pattern. You can go back and look at those for another example of a recursive algorithm, but it's a function that calls itself. In this case, each of these functions, because we have a remainder, needs to call the minimum function on that remainder. So 9 given a 6 coin, we need to call the minimum coinage for a 3. So we'd feed that back in, and then we do those calculations. And you'll see that we have to do 3, 4, 5, 6, 7, 8 calculations to get the optimal for a 9-cent coin. And it turns out to be, you give them what? 8. 8 and 1. Right. How is that recursive algorithm working? Well, the Roslin problems ask you to basically do it for any arbitrary number, but let's say we started out with 76 and now, so this is instead of making change for 9 cents, we have to make change for 76 cents. So what is the optimal way to do that? This is what the recursive algorithm is doing. It says, okay, 6, 5, and 1. Then we're going to have to call that function on 6, 5, and 1 again, and 6, 5, and 1 again, and so on. You would, right, there are a lot more calculations to do on this tree, but already something that you can see that is not very efficient is that we had to calculate the minimum coinage for the number 69 6 times already. So that should tell you that maybe the recursive tree, although it will get you the right answer, it's not going to be a very efficient way to compute it. If we did the optimal coin combination for 30 cents change, or yeah, I mean, that will fall somewhere lower in the tree. You're going to have to compute that trillions of times because this tree already, you know, we've only gone from 76 cents down to 69. And already we're doing 3, 6, 9, 12, 15, 18, 21, 24, 25, 27 computations just in the 1, 2, 3, 4th level of recursion. Okay, so this is kind of like our brute force method, going to become computationally intractable pretty fast to, but it would give you the right answer. And so this program was actually solved by this guy, Richard Bellman, who is the one who sort of invented the name dynamic programming. If you go and watch these videos, because just like these problems are continued in Roslin, they're also in the textbook. You can go watch videos from the textbook authors if you want. There's an interesting story about why he called it dynamic programming, and it was basically to help sell his funding to Congress. With sort of a benign name for what he was actually doing, which was trying to solve logistics problems somewhat similar to the Manhattan tourist problems, except for, I think it was military logistics. Okay. So effectively, this question comes down to, can we make the recursive algorithm faster? Is there a way to know that and to get to the right solution in the minimum number of steps, right? So we don't have to recalculate the recursion for given values lots and lots of times. Okay. And so his question was, instead of the time-consuming calls that over and over sort of recurring the call to the function, could we just calculate all of those things one time and then look them up? So, right, that would be here, adding 1, you have to do that all the way up until you get to 5 because that's the next denomination. So the way to get to 5 is either add 5 single coins, or really the more efficient way is to just add 1 5-point coin. So the value for the minimum number of coins at 5 is 1. Same with 6, right? Because the denominations are 6, 5, and 1. For 7, right, you need a 6 and a 1. For 8, you need an 8 and a 1. Oh, sorry. That was actually. Oh, you need a 6 and 2 1s. That's why it's a 3. Trying not to confuse you. Right, and then so how many do you need for 9? Okay, we've already sort of solved that. You need 6. That's your biggest one. And then 3 ones. I think I answered that wrong earlier. I don't know why I think I introduced an 8 value coin or something. In any case, this is how you can make the recursive algorithm faster, is by computing all the recursions first, or all the potential values for money, and then what's the minimum for those, and then you can look them up in the table. Right. So, yeah, this is what I was talking about a minute ago. I had the slides, forgot which order. So he was actually doing this math to the Secretary of Defense, and then there's a little quote there from him about why he picked the term dynamic programming. It actually doesn't have anything to do necessarily with like the kind of coding programming that we talk about. It's really programming in the sense of programming this algorithm. Some kind of weird use of the word programming. So how does that apply to our Manhattan tourist problem? Well, we know we need to get down to this point going only south or east. So this is what it kind of looks like if we were going to translate that into code. If both of the branches are 0, we're going to return a 0 because basically we're up in we're down in the corner. But if they're non-zero, then we can move south or east. So it's going to be a move on the X or Y axis. Basically, we'll treat X as moving south and Y as moving east. And that move is going to be the weight of the edge. And we already talked about how that is, in our Manhattan tourist problem, the number of attractions you would see on that block. Okay. So let's apply that algorithm here. And so instead of doing greedy, we're going to actually figure out what's the value at all the nodes. So here, right, all I've done is fill in what are the values for all possible paths here. On the internal nodes, right, filling in the possible paths requires saying, okay, how do I maximize that based on the connecting values, right? Which one is bigger? So in that case, 3 there was bigger than 0 there. And now here, to fill in this one, I can either go from 5 or 10, so that's going to get a 10. And then we basically just go through the whole thing and do that. So every circle we need to fill in, we figure out what's the maximum we can assign, right? 5 plus 2 is bigger than 4 plus 2, and so on till we fill in the whole matrix. And so check it out. Whoops. The maximum value we get by using that sort of dynamic programming where we fill in all the boxes is 34. The question is, how did we get there? Well, that's why if we were actually coding this, we would record in our list where did that value come from, right? So 34 came from this node, which is at X equals 4 and Y equals, depending on how you format your matrix, could be Y equals 0. Okay. And so if we track all of those steps, that is now the optimal route to visit the maximum number of attractions. So that is our dynamic programming solution. So Saul Needleman and Christian One are the guys who sort of recognized the translation that I alluded to earlier, where this Manhattan tourist problem is the pairwise alignment problem. If we just take our two sequences and make them the rows and the columns, we can use dynamic programming to find the optimal pairwise alignment. The only adjustment is allowing the rows and columns to shift by entering the dash marks, okay? All right, so this is already telling you how we're going to move through the matrix. And so here's the Manhattan tourist problem, except for the sequences that we had earlier. So in this case, I've sort of added in red arrows to show how we move through the matrix. And because we're not, when we get a match, we actually want to go to the next node across the sequence. So we get a match, right? A, we need to move over to T and compare that one. If we get a match there, we move across. Now, here, there's no match, right? And so the best matches are going to be the G there and there. And so this figure is all, I always think, how can I simplify this? But because it's so busy, maybe I should take out all the gray diagonals. But what you see is, like we did for the dynamic programming solution that I showed for the Manhattan problem, we've basically filled in the best solution in all of the cases, right? We took this A and said, okay, I'm going to write a red diagonal arrow every place that it matches. And so, right, when I was talking about brute force, I was saying, you know, we can't really make all possible combinations and do a scoring, except this kind of gives us a way to do that, because this matrix, all these red triangles show us all of the possible matches. And so those are the things that we want to score with a +1. And now it's just a matter of seeing, okay, how do we get across from this corner to this bottom corner and pass through the most red arrows, right? Get the most matches. This looks a little bit like that min coins problem, okay? But it's actually how you would set up the program for like a recursion to give you the dynamic programming solution. So if the sequence at I-1j, and that's just, right, you can think of rows and columns as I and j. If the weight of the edge going down into that, we're going to give it some score, weight of edge, right? And we're going to weight it differently if it goes to the right. And finally, we're going to give it some different weight if it's a diagonal, right? That's the match. Okay. In the scoring systems that we've talked about so far, the only thing we're scoring is the matches, right? We're giving them a +1. Everything else is a 0. So everything else is gray except for this last alternative where it is a match. So that's basically all I always said there, right? It's going to get a 0 for the edge weight if it's not a match, +1 if it is a match. And we're going to, the reason I'm talking about that is we're going to talk about different scoring systems in a minute. And then we need to add the backtracking pointers for the longest common subsequence, right? That's just like the Manhattan problem. Where, if we're at this node, where did we come from? Because ultimately we need to get down to this corner. And if you do that for these two sequences, you say it's really messy down here. So from this corner, it could be at any of those, right? So that's arbitrary. Okay. So not so surprisingly, there are multiple paths here. The longest common subsequence is up here, right, where the path is pretty clear. There's only one solution on those. And, you know, if any of you have done sequencing projects before where you had to do alignments at the end, a lot of times when you start to see this kind of stuff where you've got multiple indels that are close together, you often wondered if that's the right alignment. And really, there's no way to be certain. It is giving you in some cases, arbitrary fit of things that don't align well. So this is our primitive scoring where we did matches and we got a +1. And like I said, there are lots of ways that you might score this. Base pairs. When we think about how we actually want to score DNA sequences, right? Pretty clear that we might want to do all of the matches as a positive score, but then how you actually weight the mismatches can vary. And it depends on how much biological reality you want to build in, because for all the biological reality that you try to build in, you are making assumptions about, you know, how often does deamination of the vaccine lead to thymine, right? So you might want to, because that's a pretty common mutation, maybe you don't want to weight that one so high, okay? In this scoring matrix, we're going to weight all of the mutations, all of the mismatches equally, right? We're just going to give them mu as the weight of a mismatch, and then this lowercase sigma for any insertion or deletion, right? So mismatches we'll give a different weight than we give for indels. And in this case, right, I've given -2s for the indels and -3s for the mismatches. And in that case, this alignment gives a -8. That's the scoring matrix that I used, just appeared in the wrong order. So that's an example how you might do it. There are many more sophisticated scoring matrices, even for nucleotides. I'll talk about some for protein sequence in a second, but for DNA, like I said, you might want to make transitions where it's an A to a G or a T to a C, penalize less than transversions, because we know those mutations happen more often. Okay, it's just part of the biochemistry. So scoring those separately sort of makes biological sense. A mismatch between a T and a C should not be penalized so as much as a T to an A, right, or a C to a G mutation. The other one that's really obvious, especially if you're doing protein coding sequences, that you might want to weight it by position. So the cartoon that I'm showing doesn't account for the position, but if this is a nucleotide sequence that codes for protein, you might want to divide it up into codons the best you can, and then third positions, because they're highly redundant, are often more variable. So you might want to give less weight to mismatches in the third position. Okay, all of these coding systems will right, change what this matrix, and I didn't draw in the numbers because it just gets so messy, but right, the matches are still +1, but when you get down here and you see there's a C trying to match to a G, you might want to weight that more heavily. except if it's third position in a codon, you might say that C to G makes no difference. It's still a whatever amino acid. If it codes for the same amino acid, right, that mismatch doesn't do anything to the protein sequence. So I actually don't want to penalize the score of this alignment very much for that move, right? So you might, in fact, even move and count that as a diagonal, because it's the best way to go from that node to that node when you're recording backtracking pointers. Okay. And then you could also take the genome frequency into account when you're setting up this kind of matrix, right? So if you're in a super AT-rich genome, or maybe it's not usually a single genome, but in a group of organisms that have AT-biased genomes, for example, or transposable elements, I think, tend to have AT bias, you know, so if you're trying to align those, you might want to penalize AT mismatches less than CG mismatches. So having a C or a G in there, you might want to penalize that more because it's less likely, given what you know about those particular sequences or those particular genomes. Now, something to note, especially for people who are commonly doing alignments for things that are not closely related, is that DNA has inherently less information than amino acid sequence. Okay, and that is because DNA only is a 4-letter alphabet, right? You'd only have A, C, G, and T, so there are only, right, 3 ways where you could have a mismatch. So that's just inherently less information than in amino acid sequence, where you have at least 20 amino acids. So there are 19 possible mismatches and only 1 match. That's way more information about the identity of a given position. So if you're working on things that are more far evolutionarily distant, it's always better to be working on amino acid sequence if you can. Okay. So if for example, you've sequenced DNA and you know it's protein coding, translating it to amino acid and then doing the alignment based on the amino acids is going to give you more information about your alignment. So just a few notes about amino acid alignments. There are lots of scoring systems here that I'm going to talk about. The 2 most common one, which is PAM and BLOSSOM. PAM stands for Point Accepted Mutation, was originated by Margaret Dayhoff in 1970. This, I forget. It was mouse. I was trying to think of the genes. Mouse and human. But in any case, she did these alignments and then computed what the proportion of changes between any particular amino acid and all the other amino acids. So this is empirically, how often do you see one amino acid shift into another amino acid or mutate into another amino acid? So that is the PAM1, or the sort of first order matrix of how likely it is for one amino acid to be mismatched with another amino acid. Okay. PAM is treated as a Markov chain, so basically a second order PAM matrix. So PAM2 is PAM1 raised PAM1 squared. So you just square all the numbers in the matrix. Okay. The important thing to note, right, if you are making the numbers in the matrix smaller and smaller, right, you're making the weights smaller and smaller, that means the mismatches are not penalizing it as much. And so why would that be good? If the sequences you're aligning are more distant from each other, then you might not, then you don't want to weight the mismatches as highly. Okay, so bottom line there, higher numbers of a PAM matrix are better for scoring things that are more distantly related. Okay. Because of that, and because, right, this whole scheme of, like, I'll just continue to raise it to a higher power, the more distantly related I think they are, it's a little bit arbitrary. And so people tend to only use PAM for things that are closely related, right? Because the PAM1 is the only one that is based on the actual data, okay? And then just a sort of technical note there that the PAM matrix is usually the log 10 of those probabilities. So that's why if we actually look at the PAM 210, it actually has these negative values. Those are really the log. Okay. And so, right, the really positive scores are the matches. Right, so how long does cysteine stay cysteine? You want to give that a really high score. Apparently, serine is, I guess you might say, somewhat volatile, right? You don't weight a match of serine very highly. And so the diagonal has the highest scores. And then things that have the same polarity, same hydrophobicity, tend to be mutated for each other or substituted for each other more often. And so you see these clusters of positive numbers that represent amino acids with similar function, similar characteristics. Okay. And then as they become more different in charge or polarity in any of those functional consequences, the weight goes down. So you're going to penalize that mismatch even more because it's going to change functional characteristics of that protein. And so incorporating this kind of scoring matrix into that dynamic programming is going to fill in all those nodes to give you the optimal solution. But now that optimal solution is really heavily determined by what scoring matrix you used. The alternative to, I just highlighted a couple examples there. So this is all the things that I was saying. The alternative to PAM is the BLOSSOM block substitution matrix. The Henikoff and Henikoff back in the early 90s. Again, I think it was all mammal sequences, but they took highly conserved regions from well-studied protein families. And they computed the transition probabilities just like Dayhoff did for PAM, except their distance matrix is, so for example, do I give examples here? Yeah, so the BLOSSOM 60 scoring matrix or BLOSSOM 62 are the percent of identical amino acids necessary to be included in the calculation of that scoring matrix. Right? You take all these sequences and align them, and you get a percent of matches in that alignment. Okay, and that alignment is using well, actually, it's sort of iterative, but let's ignore the sort of preliminary steps. In that alignment, if you have if what you have included is only 10% identical or better, that would be a blossom 10. If it's 99% identical, so these are the like really strictly conserved sequences, that would be blossom 99. Okay, so all of the blossom matrices, no matter what number you pick, are in some sense directly the transition probabilities are directly taken from sequences that have that percent identity in them. Okay. Hopefully that makes some sense. I've given you a citation here. There's some interesting backstory there, and that apparently the Hennicoffs made a mistake that actually causes their scoring system to align things better than if they had done it exactly the way they said they did it. But interesting historical note, I would say. And so that I would say, because I think Blossom is the default on BLAST, it's probably the most common, most commonly used scoring matrix for amino acid sequences. Now, that's partially because since it's the default on BLAST, and most people BLAST without really knowing what they're doing. They use Blossom 62, whether it's really appropriate or not. And the thing I have in blue here is note that Blossom is exactly the opposite of PAM in terms of the numbers. So the higher PAM score was better for scoring distantly related things. The higher the Blossom score, the better it is for closely related things, right? Because the Blossom matrix for highly conserved sequences, that suggests they're more evolutionarily closely related. And so that matrix of distances should represent closely related things better. Okay. So sister species, Blossom 62 or higher is what you should be using. And when we talk about BLAST next week, I'll point this out again in terms of how you modify that in practice. Here, right, we've already seen what's going into this, but now let's translate that into something including scoring matrix, right? So all I've done here is taken our edge weights. So moving south or east is like adding a insertion and deletion. So we weight that with a sigma. If it's a match, moving diagonally, it's a +1. If it's a mismatch, it's still going diagonal, right? Because we're still moving across the sequence, moving across both sequences, but we're scoring it as a mismatch. Okay, and it's those sigmas, the blues and the green arrows, that have our gap penalties. And so gap penalties are really important and something that I guess in my experience, people don't give enough thought to. So just as an example here, which of these alignments is better? Right, the only difference in what generated these two things is what was assigned as the gap penalty. Okay, and I think for this one I modified the open and insertion, or open and extension, which we'll get to in a minute. But right, this one made it easier to insert a new gap. This one made it expensive, so it penalized it a lot to open up a new gap or start inserting dashes. And they give you very different answers, right? Now, most people would probably consider this to be the better alignment. Again, going back to the longest common subsequence problem. And because when you get an extension this long that matches, that's pretty unlikely by chance, and so it might actually represent some biological meaning. Okay. And so studying sequence like that sort of gets to, and this is, I think actually this might be an example of a homeodomain, but the idea here, homeoboxes are just a good example where you have these short regions of highly conserved sequence in kind of an ocean of mismatches. Because if you compare the homeobox region or homeodomain containing gene regions of fly and human, there's tremendous conservation actually in the gene content. But if you did we call it global alignment, right? An alignment with low gap penalty, you get something like this, where it doesn't look like they align at all. And this is why even when we started getting sequences for these things, it wasn't recognized where the similarities were right away, because this kind of solution is not trivial, right? Knowing how to weight gap insertion and extension. Okay, so this is a local alignment, even though it makes it look like the global alignment is pretty bad. This is a somewhat more global alignment, but the local alignment is probably telling us something functional. And so how do you go from global to best local alignment? This is I took out all of the arrows here and just showed the best path for those two solutions. Okay. If you were doing the Needleman-Wunsch dynamic programming solution, you would get this one. This is the best solution. But this is sort of the optimal solution in terms of functional information. So how do we get there? The global alignment versus the local alignment. Well, that's where the Smith-Waterman algorithm comes in. So they basically just decided. They said, well, what will happen if every time we sort of go negative in our score, we just make it 0, okay? And then we pick up at the highest value of scoring that's left in the matrix, okay? So the way the algorithm works here, NW is Needleman-Wunsch. So like I said, the fourth option, the cell that would give negative values are all just scored as 0. And then when you hit a 0, you look for the next highest value that's in the table rather than the bottom right cell. So you're sort of looking for what's the new highest bottom right in a sequence that I haven't already aligned. Okay, and so you do that over and over again every time you hit 0. And so that gives you this kind of thing. That's the global alignment in that region, but there could be potential other local alignments that are similar. Just happens to not be the case for these two sequences. Okay. All right. So you start tracing back, you hit 0, you look for the highest value, make that your new sort of bottom right, start tracing back till you hit 0, do that over and over again. And what it does is chops this huge alignment into lots of little local alignments. And then you're basically looking for the local alignments that have the highest score. So like I said, a big problem for local alignment, well, for alignment generally, but particularly local alignments, because, right, the difference here is when we go negative, we're going to make that 0. Well, the fastest way to go negative is to have a high gap penalty. And so if you want to break up your alignment into lots of little local alignments, having high gap penalties is a good strategy. Okay. And although I've mentioned gap open and gap extension, up to now, everything we've done just had sigma, so it was minus sigma as the gap penalty. But you know, in real biology, these two things, these alignments are essentially equivalent, right? You're going to get a TC matching to an indel in both of them. The question is, which one is correct? Okay, and that is going to depend on the gap penalty. Because of the way mutation works, we would want to somehow come up with a scoring system that would tell us this is the optimal solution, right? Because this one requires 2 mutations, and this one only requires 1, because you could get the, you know, a 2 base pair deletion is sort of more parsimonious than getting 2 1 base pair deletions, right? Because there are fewer mutational events. Okay, and way back to the beginning, I mentioned how no matter what scoring system you use, you are building in assumptions about the biological reality of what goes on at the sequence level. And this is a perfect example. Right? Most people would agree that just based on parsimony, 1 mutation instead of 2 mutations is the way we would want to score likelihood. So, not long, actually I don't remember if it was in Smith-Waterman, but in any case, the development of different gap penalties, one for opening and one for extension, are what's in every alignment program now. And so you have to decide how much are you going to penalize a new indel versus penalizing extending that current indel. Okay. And typically you're gonna, the default on all of them will be that it's much more costly to start a new gap than to extend a current gap. And I think this is the last slide. Right. So just a couple notes to keep in mind. These will be important, particularly when we start to do BLAST. And I put it in black down here, the sort of bottom line that if you ever have to think about, well, I have no idea what my gap penalties ought to be. Usually software, either if it's the manual or BLAST, it'll be in the available options. They'll give you different preset gap open and extend penalties to choose from or make recommendations for what you use. But in principle, because insertions and deletions tend to be highly deleterious, the more closely related two species are, the higher you want to penalize insertions and deletions, right? Because they're very unlikely to occur, right? That's because they're deleterious. And so when you're aligning two things that are closely related, you want to penalize creating an indel because you think it's unlikely that would be real. Okay. The more distantly related things get, the less you want to weight that penalty, right? Because indels do happen. Okay, and then bottom line, sometimes getting the best alignment is a matter of trial and error. If you get alignments that look like this, and you're convinced that there must be something in there that should align, then you can rerun it with different gap penalties or a different scoring matrix, right? You can make the blossom lower, right? If, for example, if you're using more distantly related things, or you think you used too high of a BLOSUM matrix, and so it's penalizing the mismatches too much relative to gaps, for example. Okay. So that's it for today. First time in a while, no coding assignment. Okay, well, in class, we'll work primarily on making sure everybody is able to get on Exceed and able to transfer files to Exceed using BLOSUM. Okay.